All right, good morning, everybody. I feel like we left off with penicillins. I'm pretty sure we didn't move ahead to that. So as I said before, when we're thinking about all of the plethora of different antimicrobials, the real idea is to think about the specific targets as far as the antimicrobial agent goes. The respective names, I don't think you really should get bogged down with all of those details, although the next handful of slides kind of bogs you down with those details. But we're going to kind of just gloss over some of that. So we did talk about penicillin and the respective ring structures that we see once we kind of modify or tweak it with some biochemistry. We potentially can impart some different characteristics, maybe some advantages. And down at the bottom, one is a possibility from resistance to the beta-lactamase enzyme to keep it from chewing it up, all right? Not always the case as we look at this. And there are other microbes that have different mechanisms rather than beta-lactamase that gives them an advantage to degrade that. The next ones, as we look at these are dealing with the ribosome interacting in a handful of different ways to basically stop protein production. So protein synthesis inhibitors targeting different regions of the ribosome, the 50 or the 30S, and then, as I said, ultimately stopping the synthesis of that. And that dramatically drives a wedge in the normal day-to-day functions of that microbe, interfering with all of that and then basically causing its demise. The metabolites, the sulfonamides, trimethoprim are anti-metabolites, basically blocking stuff like folic acid synthesis. I think we described this what day around Monday. We described the fact that folic acid is something that we're picking up from our diet. We're not synthesizing that on our own. Microbes are synthesizing it. So if we can drive a wedge that way, that definitely doesn't have issues when we think about maybe side effects as a result of that, because it's not something that we're producing. And then others, purine or pyrimidine biosynthesis also inhibiting the synthesis. This next image is really thinking about, well, really shifting over from trying to target bacteria to thinking about stuff like fungus. And there are all sorts of details with fungi that we'll start touching on a little bit later when we think about some of these as pathogens. When it comes to fungus, it is extremely difficult to try to combat these respective organisms. They are eukaryotic, like our own cells. And as we showed some examples with the antibiotics targeting bacteria, there are some bonuses with that because like maybe the folic acid synthesis, it's not something we're doing. But trying to treat a eukaryotic cell where we are also eukaryotic sets the stage for some side effects, some illness as a result of the drug. And in some instances, trying to treat a fungal infection, patients describe the treatment as potentially being worse than the actual infection because it takes so long and potentially causes some serious damage. Depends on the type of fungus, all right? So as an example, as we're looking at this, something that is found in our cells in the plasma membrane, we see cholesterol molecules. We see cholesterol elsewhere as well, but in the membrane, cholesterols are there. It's important to lend to some of the fluidity of the membrane. In a fungal cell, it's not cholesterols, but ergosterol, a close cousin, if you will, at the molecular level, all right? And we can try to target some of this, but it also can lend to some issues with our own cells because of kind of the similarity with the molecular structure, all right? It is found in the cell wall, and fungi have cell walls, we don't, right? So there are some potentials to think of it as a target, but it really kind of, again, depends on the circumstances. There are other things that we can think about as actions or classes of drugs that target elsewhere, all right? So this, we're looking at maybe a yeast, a fungal cell. We may inhibit DNA or RNA synthesis with flucytosine, the microtubules with griseofulvin, and inhibiting mitochondrial with naphthoquinone. There are, back to this, here's our ergosterol within the membrane of our fungi. We would see cholesterol if we were looking at our own membranes up there, all right? So we can target some of this inhibiting the synthesis of it. A couple of different drugs, azoles, imidazole, and there are a boatload of other azole drugs, right, that we will see great antifungals. We'll also talk about some of the protists, some that are also eukaryotic. Some of the drugs that we talk about for treating fungi, we also think about as a potential to treat some of these protozoal infections. There are definitely some similarities and overlaps with that, all right? Membrane, we can see some polyene drugs that interact with that, disrupting that. The synthesis of chitin, which is like in a plant, cellulose making up the cell wall. Chitin is what we see in fungi, giving us a very tough cell wall to protect them. So polyoxins and nicomycins, both of these interact with the synthesis of chitin or inhibit the synthesis of that. And then some other beta glycans inhibitors with that. So multiple ways that we might be targeting fungi, and it also kind of depends on the type of fungus in the first place. Here we've got some yeast cells infecting the lungs. Pneumocystis jirovecii is this organism. I kind of like this image. I don't like the organism, but I like this image because you can see it's like that picture that we just showed with the yeast cell with the thick cell wall. You can definitely see that with these guys. Very thick cell wall, the nucleus right in there. That's a pretty good image right there. So in these cases, we think about some of the ideas that we just discussed, trying to treat that. This pneumocystis is something we will describe a bit later on as an opportunistic situation. It's not something that we think of as seeing normally, but if you've got a compromised immune response, if you're battling with HIV, it can be something that is a pretty common occurrence. And that also raises a flag. If we start to see this in some patients, we start wondering other things, what's going on. you potentially already know what's going on, and then you detect this. But this might be one of those first instances that you recognize issues. So shifting and thinking about virus, acyclovir is an analog of guanosine, and in turn, this gets in and interacts, driving a wedge in the cycle that the virus needs to interact and ultimately replicate. So this binding to viral DNA polymerase and basically stopping the formation of it, so it can't synthesize its nucleic acids, and that's basically all of the details as that's going through there. There are more ideas as we look at this antiretroviral therapy as handfuls of different targets, and when we think about trying to target or treat HIV, it's oftentimes a multi-drug or cocktail approach where we're trying to throw as many different things at the virus as you possibly can, trying to inhibit its production. And I think we have made some pretty serious advances in treatment and therapies. See all sorts of things talking about people that ultimately get to the point that they're undetectable. They don't have any hints of having that, and in turn, the potential for them to transmit it is basically zero. Although on the commercials, you don't see them saying that exactly. They don't want to make those claims, but it's pretty likely that these individuals don't have any chance of transmitting that, and I think that is impressive. I've seen some other stuff, I haven't read details about it, but other potentials with some people that maybe have been cured. I have heard of some other stuff with like bone marrow transplants and some individuals that have seen similar situations. I think that's interesting, right? There are definitely some genetic ideas behind some of that stuff where there are individuals that we might classify as being super immune. People that never get sick, right? Which I think is pretty crazy. I wish I was in that category. I think I've got a pretty good immune response, but not something that I would say I'll never get ill, because I know I will. But there are others that may say that's where they're at. And in turn, they may potentially harbor some genetics that allows them to truly be resistant to all sorts of things. HIV as an example. I wouldn't go out and test that, right? Just to see. So now we want to shift and think about the opposite. Instead of trying to target and kill these microbes, flipping it around from the microbe perspective and think about how do they deal with what we're trying to combat against that. So there are all sorts of ideas where we could see some resistance. And with this, we may see some microbes that harbor a handful of these potentials. Maybe something that we might call a super bug or a super pathogen, something that is highly drug resistant, having multiple things in its arsenal to in turn ensure its success, to ensure that it will go on to infect another day, right? So there are some, as we look here, this efflux pump, where some cells having this, if, as we see a handful of different drug names up here, they have the ability to just pump these suckers right out. So that antibiotic comes in contact and the bacteria is like, oh, and I'm going to shoot you right back out of there, pumping it out, never giving it enough time to actually do what it needs to do inside of that cell. It just ships it right out. There are others where we may block the penetration so that the specific agent doesn't have access to get into the cell in the first place. We either pump it out or keep it from coming in to begin with. There are handfuls of different enzymes that can inactivate these antimicrobials. We talked about the beta-lactamase as an example that will chop it up, chewing up the penicillin or its derivatives, and a handful of others that might be targeted by some of these enzymes, destroying or degrading them. And then others that will interact by target modification, preventing like the drugs inhibiting the ribosome or the protein synthesis. It basically makes it so that that doesn't happen, and we're able to continue to infect. And in some instances with virus, that gives them a serious leg up. So we showed in the last chapter, and I kind of feel like I should have stole that picture and put it in here, the plates where we can see the little discs, the antibiotics on that plate, and then seeing stuff that's dying, right? The big zone of inhibition would mean that's a potential drug of choice. In those circumstances, when you've got it on a plate, you've got a specific, you've got this lawn of growth, but you've got a specific amount of antibiotic that's on that little piece of filter paper. And it may be effective, it may be ineffective, right? The question might be, what's the dosage? How much do you truly need to prescribe or administer to see an effect? Just seeing that on the Petri dish may clue you in a little bit, but you really don't know concentrations, all right? If we think about a lab test versus a human, what we're going to be giving us is different than what you put onto that plate, right? So that opens up other ideas where we can actually quantify. We could do some dilutions and see what's going on, all right? So in this dilution test, we're not diluting the bacteria, all right? We grow up a big flask of bacteria and you've got a boatload of organisms in this flask. And you put, whatever we've got in there, looks like a couple milliliters, right? So you would pipette the same amount, the same volume of liquid, and within that liquid would be the bacteria, and roughly the same amount of bacteria would be pipetted into each of these tubes, right? They all look nice and turbid, all right? The dilution is the antibiotic, not the bacteria, all right? And as we dilute the antibiotics, and that's what we're seeing here, from highly concentrated antibiotic to pretty dilute antibiotic, all right? This MIC, or minimum inhibitory concentration, gives us, like I said a second ago, the ability to quantify, to look at this stuff and know specifics like the dosage that's needed to kill these bacteria. So when you're trying to figure out the MIC, or the minimum inhibitory concentration, we're looking at these tubes. It's truly just a visual thing. And we can see turbid, and clear. And so the idea would be, whatever this dilution is, the first one that's not turbid would be our minimum concentration that's effectively killing them all right? At least visually, all right? Could there still be a few cells in this pretty low concentration that are viable? Maybe, right? But at least visually, it looks as if at that concentration, that, it's no longer turbid. So we say here, 99.9%, it's like Lysol, right? What is that 0.1% that you got to cover your bases, right? Basically kills them all right? But there could be a little bit that's maybe still within there, all right? We mentioned before stuff like Staph aureus and thinking about, swabbing all of our bodies, growing up the staph that's on our bodies and testing them just to see, right? It's likely that we would find differences, like different types of resistance. Some antibiotics would be effective whereas another person, they might be ineffective. We also could see issues like this. If we were to do an MIC against all of our staff, we would see, even if it's one antibiotic that seems to be the drug of choice for all of us, there's going to be some variation as far as the concentrations that we might be seeing, because it is, each of us would have our own separate population of these staphylococci with some subtle variations, all right? You would kind of expect it's going to be pretty close to the same ballpark, right? But there might be a little bit of variation, all right? Definitely some variation with the respective drugs that you might be doing. So this is basically the same idea as what we just looked at, except we're doing, instead of like test tubes full of bacteria and a respective drug, and you probably, although all we're showing here is this, you'd probably have a rack full of these and you're testing multiple drugs simultaneously. That's a lot of stuff. That's not really that big of a deal, but it's a lot of tubes, all right? The micro dilution gives you the opportunity to have a lot of tubes, but they're not all test tubes. They're little micro wells holding microliter volumes instead of milliliter volumes in those test tubes, all right? And the idea with this would be an opportunity to test a specific microbe against a handful of different antimicrobial agents simultaneously, giving you the opportunity to figure out the MICs with each of these drugs, all right? So looking at this, the antimicrobial, the drug goes, and there you can kind of see it, this micro titer plate across the top is numbered 1 through 12, and then down the side A through H. So each of these rows, as we look through here, lettered A through H, and the columns 1 through 12, we're looking at one drug in the respective well, or row, I should say, and then diluting it from 1 through 12, all right? So at this, the drug is, it says here, susceptibility test, low concentration, high concentration. We got a lot of the drug here and very low over here. I kind of feel like it should maybe be flip-flopped around, high concentration to low concentration, but that's okay, it doesn't matter. As long as you know which is which, all right? So as we look at this, and this would be like a serial dilution, so twofold dilution, something like that. This concentration, really low antibiotic. Here we've got pretty high, all right? And at this point, 32 micrograms per milliliter is this concentration, and we keep getting more and more dilute. So the highest concentration at this point, we are still seeing growth with our clindamycin, all right? All of these little bits are the bacteria. You can see your control down here. This is showing you growth. So that tells me, right as I look at this very first row, clindamycin is not what you want to use, right? If every single one of those has it, and I can look down and see others that don't, it's pretty apparent that clindamycin would not be our drug of choice. And we say here, well, this MIC's got to be greater than 32 micrograms because that's what we put in there was 32 micrograms, and we don't see anything. Could it be completely resistant? For sure. But to know that, we would have to continue with another plate and start diluting at our first one would be 32, and we would keep going from there, right? So as we do that, or keep getting more and more concentrated, as we do that, maybe we would find the dosage that's actually effective. But by looking at this, it seems to me like it's not, all right? We could see some other stuff going on here, and this is some oddities. We'll mention this here in just a second, all right? And it's not telling me what we've got going on right here, but it's showing some of this other stuff, all right? I'm assuming these are some other drugs, all right? So here with penicillin, we've got pretty low concentrations at this point. We've wiped them out, and this would be our MIC. Over here, the same, and that you can see they're not all the same dosages in each of these wells. It's different drugs, different dilutions, all right? And so the hint of a little bit of stuff here with erythromycin, it's not as pronounced as this, but there's a little bit, and then here there's nothing, all right? And then we've got more that are some controls down here, all right? Up here with this weird looking stuff, all right? Sometimes, and I don't do this type of stuff. It's more like the Kirby Bauer plates with the little discs on there, all right? But you can see some stuff that's kind of similar to this, all right? There are some drugs that are kind of like on the fence, right? They're sort of in between. They may slow the growth, but they don't completely kill those microbes. And so you might see a little bit where there is some effect, and you get little, if you're looking at the plate, little specks of bacteria that are growing in that zone. So it's slowed it, but then once it continued to diffuse out into the media, the drug did, the bacteria finally are able to start growing. And if you give them enough time, they would fill in that zone, all right? That's kind of what we're seeing up here, where there is a period of time where they are inhibited and then not, all right? That's kind of my take on that, although it's not telling us any details with that. That's what I think. This, I like this. I think this is pretty interesting. And it's like the Kirby Bauer technique, the plate with all of those discs. And it's also an MIC at the same time, all right? So we've got two different things happening on a Petri dish. So this E test, there are all sorts of different antimicrobials. In this case, it's vancomycin that's on this strip of filter paper. But you could have, just like all of the little circles, the little discs with specific drugs on them, you could have similar E tests with that drug, but it's not a specific amount, it's a dilution, all right? And you can see we go from very concentrated vancomycin to very dilute, clear down here at the end of our strip, all right? You would get your Petri dish, you'd create a lawn of growth, so you'd spread that liquid bacteria all over the plate, and as soon as you've spread it on there, then you'd put your strip of paper onto the plate. You'd pop it into the incubator, let that sucker cook up for a day, pull it out and see if there's a zone or not. If the bacteria, and this is all bacteria, and this is dead, right? No bacterial growth. So if all of it is like this, well, you would know that drug is not effective. But if you start seeing some clearing, then you know that this drug is effective, and now you can get a specific MIC based off of that, not just a yes, it's there, here's a big zone. You get a big zone, plus you get a dilution as you look at it. So as we look at this, super concentrated up here at the top, and we've got a great big zone of inhibition. We've killed off all of these organisms. Looks like there's probably some writing on the underside of the plate. You've got to label your plates up, right? So that's, I think, what we're seeing right there. So this stuff, as we keep going down, it's like an arrow. It goes right into where it makes contact with the paper, and then you read that off. It looks like 1.5. That's where we're making contact with it, and we would say that's the MIC for this respective drug. Pretty cool, all right? You also can see the hints of a little bit of growth in here, right? To me, I think I might take some of that into account and think maybe I'd be looking more like this. But you're really kind of trying to think where does the bacteria touch the thing? But this is kind of what I was talking about, that you could maybe see on some of those plates where it starts, it's dilute enough that we can still kind of live there, right? And this, as we talked about Staph aureus and the methicillin resistant, vancomycin could be a drug of choice for those bacteria that are resistant, and this showing that idea. So there are definitely issues and concerns when we think about antibiotics and we think about drug resistance. Basically, shortly after the discovery of penicillin and the really kind of widespread use it for everything, we start recognizing some resistance, all right? I mentioned before, there are microbes that have resistance just normally, but if we bathe ourselves in antibiotics and happen to have some of those microbes within us, now we set the stage for a population that is different and resistance shows up. So that's an issue, all right? The fact that resistance is a big problem, all right? Producing new novel antibiotics, well, we used to think that was big business. There were lots of pharmaceutical companies that were doing all sorts of research trying to find and discover new drugs. And then lots of them throw their hands up and say, you know what, we're going to do something different. This is not, we're getting out of the antibiotic market, right? It's too costly. This is a very expensive process to go from out digging in the soil, trying to discover some microbes that potentially harbor some of these activities, to going through the process of truly identifying and recognizing whether or not what they're producing is toxic to the ones that we want it to be and not toxic to us. Those are all issues that take a huge amount of time and massive amounts of dollars, all right? So with that said, all of these problems and resistance and everything has made it that we don't see lots of new drugs coming to the market, at least not antimicrobial drugs. There's all sorts of drugs that you'll see commercials for. And if you're hypochondriac, you love that, right? You're like, holy cow, I have that, right? That's a different story, all right? You probably don't self-diagnose, right? You probably don't want that. All right. So we could look here back to the, mid 80s, 16 or so new drugs approved in that window of time. From 88 to 92, we're down to 14. As we get over here from 2008 to 2012, only 2. And I'm sure those numbers, as we're looking now, are right around that or less, right? Not so good. They know that basically a year, 2, maybe 3 years after this drug makes it to market, we're going to see some resistance. That's a whole other issue for people just saying it's not worth it, all right? And that's bringing us to these thoughts of, will we be in a world where these things are no longer effective? It sure seems as if that's where we are marching. And that, to me, is rather terrifying. That's good stuff. All right, hang on. I stopped that. I didn't even end my share. I just closed it. Sorry, Zoom. Okay, I do want, I've got a little bit of time. I do want to start this next one. It's on our schedule of stuff for this week. There are a handful of chapters that we are skipping over. Some of it going through a few more details with some of that, but things that I think truly not necessary for what we got going on. We want to jump ahead. We thought about some chemical mechanisms. We thought about some antimicrobials. Now we want to think about the immunology behind it. We want to think about our defense mechanisms, and we also want to think about how the microbes are able to potentially combat that. We showed some of the resistance as we were wrapping up the last chapter, but that's resistance as we think about drugs we're throwing at it. How is it that they can come in and resist our own response. We've mentioned a few things as we've looked at and kind of introduced you to some of the different microbes. Things like the capsule layer, the sugar coating that some of these bacteria can secrete around themselves is a mechanism to hide from our immune response and to give them an opportunity to kind of stick within the body and go undetected, lending to potentials to colonize and lend to disease. So we want to think about those ideas as we move through here. And the last chapter of this chunk of material, we'll think about when stuff kind of falls apart and we have issues with our body targeting ourselves. And that's a problem, right? When immunity doesn't work to protect us, it can be a serious issue. So as we move through here, we will think about some diseases as we progress along. And as we transition into the last unit of material, when we're really thinking about diseases, we will also kind of think a little bit about some of the immune response as well. So these chapters kind of work together as we move through. So chickenpox, we've talked about it already. Varicella zoster is the virus, and we can see a little bit here with the rash, characteristic puck-like lesions, and some of this, as we're looking at it, is part of this innate response. This inflammatory response is part of the innate immunity, and oftentimes we think about inflammation and we don't want it, right? We want to try to inhibit that. Anti-inflammatories, take a Tylenol, right? There are issues with inflammation, and there are definitely some benefits with inflammation as well. So we often think of it as something you don't want, right? But deep down, you kind of need some of this in order to get these immune cells motivated to do something about it in the first place, right? It's kind of a signal saying, we got something going on here, right? More than just like, I need some Tylenol, right? There's other problems going on, all right? So it helps to stimulate cells and part of this immunity to really recognize something's going on, all right? And we'll go through and talk about details with that as we progress. So I'm pretty sure we showed some of this stuff chapters back as we were thinking about cells, but maybe I'm mixing this up with my biology class. Could be. It all goes together, right? There's all sorts of ideas when we're thinking about multicellular organisms and different types of cells. And this ultimately lends to some ideas with part of this innate response, protective mechanisms, stuff like tight junctions that are a tight seal from one cell to the next, something like intestinal cells. You want to keep all of the contents of your meal within the intestines and not just spreading out into the body cavity. That would be a mess, right? That would be crappy. So we don't want that happening, right? So these tight junctions help to ensure stuff stays where it should stay. Other cells that don't need that, we can see connected, kind of Velcro-like, with these desmosomes holding them, but not holding them sealed together, all right? And then others with the gap junctions lending to communication, crosstalk between these cells, all right? Other things, signals, chemical signals could be moving from one cell to the next through some gap junctions. So it truly depends on the types of tissues that we're looking at or the types of cells as far as some of these interactions. And it also sets the stage for thinking about how all of these cells work together to basically lend as this kind of first level of defense. So initially, at the level of our skin, this is definitely a huge part of the innate immunity. The unbroken skin provides, oh, hang on a second here. Not one person on Zoom piped up to say, you're not even sharing this stuff, right? Which kind of makes me wonder, are you guys even watching this at home? What the hell's going on? Did you run back to the kitchen to get some more Cheerios? Okay, that's all right. As long as I see your names there, you must really be there, right? Whatever. Oh, now they're like, I'm here. Ah. Okay. There we go. As soon as I'm like, wait a minute, I didn't turn the thing on so I could deal with the camera. So I see my torso. That's what you get. Welcome, YouTubers. Okay. So the skin, as we look at this stuff, like I was saying a second ago, the unbroken skin, that's the idea, all right? If we've got a wound, we've got issues. We just opened up this and have now a portal from the outside world to the inside of our body. And that sets the stage for definitely some issues. And we can talk other details about wound healing and all of that, but I don't think we want to get bogged down with that. So multiple layers of the skin, the outer bit, the epidermis, numerous layers of dead cells that are a protective barrier. We move down from the epidermis into the dermis, a very thick layer, and then to the lower bit where we see some of the fatty tissues, the deep layers, the hypodermis. So it's this top piece, the epidermis, that, like I said, the unbroken skin. As long as we have this intact, it provides a very good barrier. There are definitely some mechanisms where some organisms can get in, like through the hair shaft into sweat. We can see little pores here in the sweat gland. There are numerous opportunities for that where we don't need broken skin, but for the most part, this intact layer is a good mechanism for that. This one, oh man, it disturbs me. I think it's impressive and a little bit terrifying, all right? Sporothrix shankii is a type of fungus, and this is something that gets in more than just the skin, well, the epidermal layer. We're moving into the deep layers as we move in through the epidermis and basically target the dermal layers, right? There are other things like athlete's foot or ringworm, and they're only skin deep, right? They stay up here within the epidermal layers, right? But sporothrix goes deeper, and that looks like a deep wound, right? That's bad news. And you'll never think about roses the same way. So they call it rose gardener's disease, but it doesn't just have to be roses. There are all sorts of other things where you could contract this. The fungus lives on the vegetation. It's on the roses. It could be on some shrubs, like some evergreen bushes, potentially can be some good ones. Things where you may decide, I ought to do some pruning. And you get out there and you start pruning stuff and you get some scratches. Roses have thorns, and you pretty much know you're going to get some scratches if you decide you need to prune the rose bushes. Same with some evergreens. They can scratch you up as well. And if it happens that the fungus is on that vegetation, you get a scratch, it cuts through that epidermis, basically inoculating you with that fungus. then it starts to grow and proliferate, and now you got some serious ill effects, right? It is not something that's super common, but it is something that could be found around here. There are individuals that become infected with Rose Gardner's disease in temperate regions like this, all right? There are plenty of others that I'm like, oh man, I'm so glad I live in Idaho because we don't see this stuff here, but this one, it's not the case, all right? With that, all right, through these cuts, the thorns or whatever inoculates that into you, and you get these issues oftentimes on your arms, all right? But kind of depends on the circumstances, all right? And we'll hit some of that later as we start thinking, diseases and moving along. So we won't get bogged down with all of that right now. Here we've got an electron micrograph showing some ciliated cells and non-ciliated, but really it's the ciliated stuff we want to think about right here. These little tufts of ciliated cells. So these we can see in the trachea, and basically if they are functioning properly, they basically help to flush and pull the mucus up and out of there, away from the lungs. Along with that, any garbage, any debris or microbes that are potentially stuck in that mucus get removed. So moving up the esophagus and then up to the esophagus, excuse me, and then swallowed down, potentially. So this also part of the innate response. If these ciliated cells are doing the proper functioning, it helps to flush that mucus up and out of there. Speaking of mucus, the goblet cells are cells that do the job of dumping mass quantities of mucus. So with that, we could see the microvilli up here at the top, not the same as the cilia below, but the microvilli, and then all of these vesicles, these secretory vesicles harboring the substances that's going to make the mucus mucin. The mucin gets released as these vesicles make their way to the surface, and then through exocytosis, they expel them, all right? So this, as we're looking at this blow up, this is intestinal epithelium. So we could see intestinal cell right here, more intestinal cells, and I'm assuming this bit right in here would be the lumen, right? These cells, as we see them all together like this, would have tight junctions, right? Welding or caulking each of these cells together to keep all of the stuff in the lumen, right? And the mucus secreted to the outside or into the lumen, right? Tears flush microbes away, protecting the surface of the eye. Urine, not the same as tears, but it's a similar flushing, moving microbes out of the urinary tract. And so this typically a sterile site, the urinary tract, all right, typically. But there are definitely potentials for microbes to come in and cause issues with UTIs, and we will hit that stuff later on. All right, and I think, looking at my watch, I think that's probably a good stopping point for that right now. Sorry for those of you tuning in that missed a handful of those first slides, but it's all good. All right, now I'm stopping the share.